Welcome to this week's episode of Brainstorm, where we give you a glimpse into the world of science for Wednesday, February 27th, 2013. Our top story comes from the world of neuroscience. A study done by Oxford University is looking at ways to reduce the tremors associated with Parkinson's disease. These physical tremors can be devastating, but there has been some success in reducing their effects using deep brain stimulation. Unfortunately, that involves implanting electrodes in the brain, which is a major surgery. So instead, they looked at transcranial alternating current stimulation, TACS for short. This involves simply placing electrode patches on the surface of the skull that continuously deliver a harmless electric current. It's far less invasive and showed a 50% reduction in resting tremors among the 15 patients in the study. This works because the alternating current is set to match the signals within the brain associated with tremor, thereby partially canceling it out. Although encouraging, this first study was just a proof of principle and there is still development necessary. For example, a system that could continually monitor brain waves and adjust the electrode patch's output accordingly. And without being much more invasive, the electrodes and other systems could be placed just under the skin, allowing those with Parkinson's to live more normal lives. Next is looking at the effects of long-term exposure to this current and seeing if it may even help the brain learn to regulate itself again. Next, we have an update from the world of technology, with more exciting developments from the field of robotics. Last week, we discussed robots being programmed and trained differently to work better with human teams. Similarly, another helpful function would be if robots could understand and learn human language, which is exactly what a group of French researchers is working on. In this case, the robot itself isn't new. That's just a little humanoid called the iCub. The real developments are happening in the brain of the robot. And in this case, the term brain is slightly less metaphorical because the robot is run by an artificial neural network, essentially software designed to mimic some of the functionality of the human brain, based off years of neuroscience research. It's currently a very simplified mimic of the human brain, but the results are quite remarkable. Using what's called recurrent construction, connections within the artificial brain can create localized reoccurring loops that help in learning. The robot can learn sentences with new grammatical structures, link two sentences together, and even try predicting how a sentence is going to end as it is being said. As you can see from the video that's been playing on screen, the robot effectively understands language. This functionality is beyond what can be directly programmed in with our current knowledge. The system must partially learn it. The group will continue to develop this system, which is one step closer to fully functional androids. We end with news from the world of genetics. A study by Vanderbilt University involved messing with the biological clocks of microorganisms, and the results have interesting implications. First, we need to review some basic molecular biology. Protein synthesis involves DNA being transcribed into messenger RNA, and then that messenger RNA being translated into an amino acid sequence. The mRNA sequence is made up of codons, a set of 3 nucleotides that correspond to one of the 20 amino acids. Since there are 64 potential codons, but only the 20 amino acids, some have synonymous codons, different nucleotide sequences that result in the same amino acid. But interestingly, these synonymous codons aren't necessarily equal. Research has revealed optimal codons which are one in a particular set that cells process the fastest. So the idea was that critical genes would have all their codons optimized to make protein synthesis as fast as possible. However, when studying biological clocks, the scientists tried optimizing the genes involved, and to put things simply, everything broke. The biological clock within a species of mold was completely disrupted, and within blue-green algae it shifted the organism to a 30-hour cycle. That suggests that the speed at which a protein is synthesized may have a critical effect on if it is successfully created. Not only does this give insight into the biological clocks, but adds another layer of complexity and regulation within the genetic code. As well, this discovery could have practical implications for biotechnology. The biological clock regulates many other functions within an organism, so learning how to control it could boost production of biofuels and other products. Well, hope you enjoyed this episode. If you did, please consider subscribing and be sure to check the links in the video description.